Welcome to an hour of HealthMade Radio. HealthMade is a community for natural health seekers where we educate people about common health conditions and share extensive research on the most effective natural health treatments and promote legislation that protects our health freedoms. A core concept belief is in the innate intelligence and healing power of the body, and if properly supported spiritually, emotionally, and nutritionally, it can find its way back to health. HealthMade Radio will bring information from integrative health experts throughout the world. Check us out at healthmade.co. Health is what you make it. I'm Dr. Michael Karlfeldt, and I will be your host. Today's guest is Dr. Judy Mikovits. Dr. Judy Mikovits earned a Ph.D. in biochemistry and molecular biology from George Washington University back in 1992. At National Cancer Institute, she developed purification methods for interferon alpha used in the first immune therapy treatment for hairy cell leukemia in 1986. In 1986-7, she developed production methods to ensure biological materials manufactured using human blood products were free of contamination from HIV-1. Her Ph.D. thesis changed the paradigm for therapeutic treatment of HIV. Her pioneering work during a 20-year career at the National Cancer Institute includes the discovery of the modulation of DNA methylation machinery by human retroviral infection and the development of the concept of inflammatory cytokines and chemokine signatures of infection and disease. Therapies which are still standard of care 25 years later and credited with saving millions of deaths from HIV/AIDS. In 2001, she moved back to industry where she directed the cancer biology program of Epigenetics Pharmaceuticals. In 2009, Dr. Rossetti and Mikovits Labs isolated for the first time a new family of human retroviruses, then identified as XMRV. In 2012, it was learned XMRV was a contaminant of the Silverman lab, and the XMRVs isolated were a new human exogenous and transmissible retrovirus family, which are strongly associated with neuroimmune diseases and cancer. Dr. Mikovits has co-authored more than 50 peer-reviewed publications and book chapters, and she has also written the book that is an amazing read called Plague. Dr. Mikovits, I am so honored to have you on the show, and just reading everything that you've done through your career and the impact you've had in people's lives, it is absolutely astonishing.
Well, thank you, Dr. Karlfeldt. It's been quite a journey as I hear you talk about it. We didn't appreciate much of what we knew, and we used to call things we didn't understand bystander effects. And as we learned more of the biological, biochemical mechanisms, of course, we knew we had treatment strategies right there for patients today for the gamma retroviruses, the XMRVs, that we could jump right in and treat them and prevent many, many diseases or chronic diseases of our time. So that's the excitement of that wild journey we've been on.
and I've heard you use the term gamma retrovirus. Can you explain to the listeners what a retrovirus is and what is a gamma retrovirus?
Yes, retroviruses are RNA viruses, so their genome is RNA. And the retro means they write their genome, they transcribe their genome backwards, making it into DNA. And then they have an enzyme called integrase that literally is like a pair of scissors that cuts open your DNA and inserts the retroviral DNA. It's called a provirus into your genomic DNA, into your genomic DNA. And that retrovirus, that provirus, then is expressed and transcribed whenever your cellular DNA is replicated. So depending on where it went, it's replicated and transcribed. So these viruses, retroviruses, which make up 8% of our DNA. So 8% of our DNA is ancestral retroviral DNA that's passed down from us, from our parents, grandparents, and beyond. And we think of them almost as scars from our encounters with hostile viruses in the past. So they're crippled such that they're not expressed, and they're part of our genome. So when we we name retroviruses alpha, beta, gamma, delta, based on our electron micrograph pictures of those viruses and what their DNA and their capsule looks like. So it's simply a way of describing them to fit families of retroviruses based on how they look in an electron micrograph. So the mouse-related viruses that we isolated in 2009 with Dr. Rossetti's lab turned out to be gamma retroviruses, and it's just how they look on the electron micrograph, which Kunio Nagashima, our longtime colleague, had done all of the electron micrographs of the human DNA, human retroviruses that cause disease. It's interesting that until as late as 1975, my colleague and mentor, Dr. Frank Rossetti, was told not to bother to study disease-causing human retroviruses because they didn't exist. And in 1980, he and his student and colleague, Ph.D., an MD, Bernie Poise, isolated the first human disease-causing retrovirus, which caused a very aggressive T-cell leukemia and now is associated with many autoimmune and neurological diseases like uveitis, eye diseases, inflammatory diseases, muscular pain syndromes. And so the HTLV1, which he called it at the time, human T-cell leukemia virus or lymphotropic virus, is actually a delta retrovirus. Long answer for scientific ways of classifying the viruses according to their structure and activity.
And is there one retrovirus that's worse than the other? It's alpha worse than gamma, or it really doesn't matter?
No, it really doesn't matter. The delta retroviruses are closer to the gamma retroviruses. HIV-1, which you talked about a lot in the introduction, is a lentivirus. So it tends to have is less simple, is a more complex virus, and thus it interacts with our immune system in more complex ways. So HIV, I think, was the most dangerous, if you will, the most pathogenic retrovirus of our time, and everybody will appreciate the AIDS epidemic back in the 80s and early 90s when it was, healthy young men just dying of opportunistic infections that were unheard of in a Western, civilized economy. Beautiful, healthy young men, just dying of strange infections and acquired immune deficiency, which is what HIV/AIDS means.
And the research that you did at that time was then obviously instrumental in saving millions of lives as this HIV virus was going rampant among all these young men.
Right. Yes, we did a couple of things. We worked with our longtime colleague Candace Pert, who actually discovered and promoted something called peptide T. And if your listeners want to see a description of that event in our history, a pretty good movie is the Dallas Buyers Club. So at the early part of the 80s and early 90s, Candace was looking for our own biological response modifiers, which could change the infection and prevent the cells from becoming infected, and she discovered peptide T, which was an immune modulator, a small peptide that modulated the entry of the virus into certain cells in the innate immune response. And then my Ph.D. research, at the time I was starting my Ph.D. research in 1987. I'm pretty old. In 1987, we couldn't understand why only 1 in 10,000 T cells were actually infected with HIV, and yet many, many more T cells than that were dying. So we called this, or the medical community called this bystander effects, which didn't make a lot of sense to a scientist. So as my PhD thesis, I chose to study those bystander effects, and in some part based on Candace Pert's research and what we had been doing with peptide T. My hypothesis was that the orchestrator of HIV/AIDS and the cell that we needed to protect was the macrophage. So in treating not the T cell, but the macrophage, and preventing the expression and the activation of HIV, keeping it quiet and silent as DNA and not letting that provirus be expressed and infect other cells. That was what changed literally all of treatment of HIV, and that's why in our book called Plague we talk about Magic Johnson, because his infection with HIV was announced a week before I defended that PhD thesis. So the thesis committee asked me, based on my thesis, explain will he or will he not get AIDS and die of it, because at the time people were dying literally just like flies. It was a very scary time. And so he's a hero, Magic Johnson, because he had to change the treatment paradigm. We couldn't wait to treat him until his T-cell count got below 200, or below. At the time he was exhibiting symptoms. We had to start very early once we identified the infection, and treat his immune system to stop it from being activated and keep it strong and healthy, such that he would never develop AIDS. And of course, he has not.
Yeah, we're very grateful for that you defended your thesis very well. We appreciate that a lot.
Scary time.
Yeah, exactly. We're going to just take a quick break, and then we'll be right back. You're listening to Health Made Radio with Dr. Michael Karlfeldt, and my guest is Dr. Judy Mikovits. Welcome back to Health Made Radio. This is Dr. Michael Karlfeldt, and with me today I have Dr. Judy Mikovits. Dr. Mikovits, you helped to save millions and millions of lives, and the fight was against a very dangerous retrovirus called HIV, and now we have an explosion of all these different diseases like neurological diseases, autism, cancer. My question is, would these diseases be considered contagious? Would they be considered something that is connected to these retroviruses?
Well, our work shows the answer is absolutely yes. Not to HIV retrovirus, but yes, to animal retroviruses like those gamma retroviruses that come from mice, and pig retroviruses that are parts of our vaccines and biological therapies, monkey kidney retroviruses similar to HIV. Yes, we're being exposed to and inoculated with the components, the pieces, the parts, and retroviral DNA from many, many species. In some of the technologies we're using, literally as therapies, are promoting the evolution of pathogenic retroviruses in our world.
So it seems to me, like you were mentioning earlier, that you have these retroviruses. They actually go in and they change your genetic code. So they actually change how you function, how your immune system function, how you defend yourself against toxins. And so you're driving into the system an intelligent piece of information that change your computer coding and how your computer is functioning. To me, that seems pretty risky. I mean, that's because you have no idea what's going to happen at the end, right?
Right. So it is. We are literally changing our blueprint of life at the very blueprint level, and the expression, which is modulated of retroviruses, which is modulated by the environmental toxins, such as the aluminum in the environment, the heavy metals, the mercury, the pesticides, lots and lots of things are causing the expression or crippling the immune system from being able to regulate the expression. We all know about the genes, the response genes, such as MTHFR and DNA methylation genes 1 and 3, which are critical to silencing retroviruses and regulating the expression of our metabolic pathways. So what happens with retroviruses is, and we're crippling the very machinery with GMO food, genetically modified organisms. We can consider GMOs actually as retroviruses because they're lab-made retroviruses, and they often use retroviral backbones. So even our plants have retroviruses that are critical for regulation of gene expression, and those are dysregulated by genetically modified foods and organisms.
Because they use retrovirus, or they use retroviruses in order to be able to modify the food to create a GMO food, and then when we ingest that, we actually ingest then these retroviruses.
That's correct. So we have the opportunity then to pass them through generations, and if that integration occurs in a critical gene for immune function, for endocannabinoid function, which we're seeing a lot of, our endogenous cannabinoid system, which is our dimmer switch on our immune system, extremely important for the development of autoimmune and pain disorders and many of the psychoses that we're seeing exploding in our time.
And so the different ways that we're then exposed to these retroviruses, and I assume obviously less is better than more, and the more retroviruses you're exposed to, the more likelihood you have of kind of rewriting your genetic coding that can then make it so that instead of your body functioning in the harmonious way that it should be to support health, it then starts to function in a way that is dysfunctional, which drives towards a disease. So it seems then that how are we then exposed? I mean, we were talking about GMO, but you also said inoculation. What does that mean? I mean, how are we being inoculated by retroviruses?
Well, we're being inoculated more now than ever with retrovirus-containing vaccines. So I use the word inoculated because it's the proper one for injecting. So vaccines are considered by the population or the patient population or to develop immunity. And so our vaccines of the 21st century, I'd say since 1992, have used what we call virus-like particles, have contaminants that include retroviruses known and unknown, or retroviral DNA, or components, just the proteins, and those are part of the makeup of the inoculation of the shot. So our vaccines are actually GMO. They're virus-like particles. They're not attenuated or killed or lessened live viruses or live bacteria. They're actually manufactured in cell lines, in animal cell lines, in insect cell lines, in human cell lines in the laboratory, and those cell lines, those cell parts and components are actually residual parts of the manufacturing process. So we're actually inoculating our parents, our children, anyone who gets a vaccine, particularly RNA vaccines. We're inoculating them with literally millions of retroviral, proviral DNA with the potential to make particles and recombine with the expressed endogenous elements, those viruses that we're born with that make up that 6 to 8% of our DNA, our ancestral retroviruses. So as they're expressed, they recombine, and that was and that's the horror, to produce literally new Frankenstein retroviruses that can actually be contagious and contain both animal and human viruses as a new virus. And that was the unfortunate discovery that we made in 2011 when you talked in the introduction about that the XMRV was actually cloned in the laboratory, but what we found is that it recombined with ones that were in mice and humans, and that what we isolated came from people with very real diseases, autism, cancers, myalgic encephalomyelitis, and these people had acquired these retroviruses decades earlier, or literally through their inoculations and a contaminated blood supply. So it was no different than we found the transmission of HIV in the 80s. It was the contaminated blood supply. It was through IV drug users, and at that time, we as a culture and a government mandated that susceptible populations be inoculated with hepatitis B vaccine, and we learned in 2011, much to my
dismay.
Yeah, exactly. We learned that, in fact, we probably spread HIV through these populations via the vaccination, which was contaminated with other retroviruses. And now we are seeing the evolution of these viruses. Just last week, we saw a news report showing in Canada there's a new drug-resistant HIV virus out there that is extremely deadly. And so this is all part of the same evolution in the process. And of course, we've changed our vaccine schedules since the mid-1980s, such that instead of getting 1 or 2 shots in childhood, we get as many as 80 inoculations of 20 or 30 different viruses and microbial parts. And essentially every one of those inoculations contains at least one known family of retroviruses.
That is just scary. It's really scary to see the potential, like you're saying, the Frankenstein viruses that are being created. We'll just take a quick break. We'll be back. You're listening to HealthMade Radio. This is Dr. Michael Karlfeldt, and today's guest is Dr. Judy Mikovits. Welcome back to HealthMade Radio. This is Dr. Michael Karlfeldt. Today's guest, I have Dr. Judy Mikovits. Dr. Mikovits, you painted a very scary but real picture as to what's going on and the Russian roulette we're playing when we really don't have an idea as to what the full implications are. And you're mentioning that the hepatitis B vaccine that was given back in the 80s, and you're seeing the impact of it 20-some years later. So you can't then really see the direct correlation between the vaccine unless you're a researcher like yourself to be able to study the trail, so to say. What can we do? I mean, how can we change this course? I mean, we're currently getting all these genetic materials, these retroviruses that are grown on other species cell lines, which I assume also, just like if you would get an organ transplant and then you have the immune system start to react against that, I would assume if you have something that is not even human-like and you're getting those type of cells directly injected into you, along with these retroviruses, you would have an immune system that would go completely berserk.
Exactly. That's exactly what's happening.
So no wonder we're dealing with all these autoimmune conditions that are just flourishing all over the place, where the immune system is then starting to attack the host, and we're then getting Crohn's, we're getting rheumatoid arthritis, fibromyalgia. I mean, the list goes on and on of new different autoimmune diseases that we're dealing with.
Correct. Yes. The good news. I did paint a dark picture, but once people start to see it, in the past couple of years, I've been working with various clinicians and brilliant clinicians like Dr. Dietrich Klinghardt there in Seattle, and yourself, Dr. Bigger in Germany, the Academy of Nutritional Medicine in London, and physicians are starting to see it and starting to see the related chronic diseases like intractable Lyme disease or mold illness, the brain fog, the all stages of deteriorating brain in most childhood illnesses. The doctors are seeing that. They're starting to see the connection with retroviruses. And the great news with the work that so many of my colleagues have done with HIV since the early 80s is we have therapies. We have therapies that can be used, both plant-based therapies, natural product therapies, or the actual antiretroviral drugs like Truvada or AZT, or even peptide T from my late colleague, Dr. Candace Pert. All of those drugs have been successfully used, to treat the diseases. Now that clinicians are recognizing the contribution of an overactivated immune system and the role of these retroviruses in dysregulating the immune system. So the great hope now is it's so easy to prevent. We simply change the vaccination schedules. We change. We clean up the vaccines, which we haven't been allowed to do since the law of the National Law of Vaccine Injury Protection Act of 1986, which removes all liability from vaccine manufacturers, doctors, healthcare practitioners who administer the vaccines. That law was put into place with safeguards where the Health and Human Services Department would then take over safety and efficacy testing. Our colleagues, Del Bigtree, he should be a doctor, he's good, Del Bigtree and Robert Kennedy Jr. Filed a lawsuit against the federal government several weeks ago because in their 3 years of investigation and Freedom of Information Act, they discovered that not a single safety test had been done as required by this law in more than 32 years.
So here we have then we're all being inoculated by these with these vaccines that we all think are there to boost our immune system and protect us, but in essence, they're introducing. They're all contaminated with these retroviruses that are changing our DNA, that can then make us sick just from that. And then in addition to that, it's introducing then foreign animal cell lines that can then trigger our immune system to become overactive. And so that's what they're pushing currently. And we are thinking that this is a healthy thing.
Correct.
Yeah. Is there a way then to make vaccines beneficial, do you feel?
Oh, absolutely. And so in addition to all the biological contaminants, there's heavy metals, mercury, aluminum. There are known carcinogens like formaldehyde in these vaccines, and that it's creating this perfect storm. Is it possible to make a safe vaccine? Absolutely. We know exactly, and we learned in 2011 how to clean up the blood supply. We learned from the gamma retroviruses, we learned with HIV/AIDS how to clean up the blood supply and the products. We never vaccinated an AIDS patient until very recently. And believe me, my colleagues and I scream at the top of our lungs at the preposterousness of this. So we don't vaccinate the immune compromised, and yet walk in any cancer infusion unit and it'll say, did you get your flu shot? And so we've got to re-educate our healthcare practitioners, and that's the value of a show such as this one, to say, what do you mean? Because many of the clinicians coming out of school aren't being taught what we learned in 2009, aren't being taught the valuable lessons we learned from HIV. And some of that is political, of course, and some of that has to do with just plain corruption surrounding this law. So we need to work with, advocacy in our politicians to stop these mandates throughout our states for these vaccinations and demand that the law that provided for the safety and efficacy of these inoculations be followed. And I work every day to do that, as do many of my colleagues.
Yeah, we so appreciate your untiring work and effort in this area. Really, really appreciate all the research with your background. It's so helpful for us novices in this area. What I really like to understand, just kind of so the listeners have a feeling for what these retrovirus can do and how we can deal with them. So let's look at cancer, for instance. We, 1 out of 2 is to be diagnosed with cancer, and 1 out of 4 will die from cancer. So it's a disease that is just exploding. How can you tie a retrovirus to a disease such as cancer?
Well, as I mentioned in the beginning, the very first human disease-causing retrovirus isolated by Dr. Frank Rossetti and Dr. Bernie Poise in 1980 caused an adult T-cell leukemia.
So is that the only type of cancer, or can it then be implicated in all types of cancer, or just in a very small subset?
Oh, no. In prostate cancer, breast cancer, non-Hodgkin's lymphoma, chronic lymphocytic leukemia, mantle cell lymphoma, ovarian cancer, kidney cancer, colorectal cancer, bladder cancer, hairy cell leukemia, for which we treated in the very beginning. The cells literally look hairy. So we understand, in fact, the XMRV that was cloned from prostate cancer. And so the ones we isolated were family members, were cousins of that clone virus, and so these viruses are associated with the kinds of hormone-related cancers and organs that are exposed to the environment and to the toxins, such as the gut, such as the kidneys, the bladder, the prostate, breast, ovarian, hormone-driven cancers. These viruses are hormone-driven. That's what drives them to be expressed. So many, many cancers that once were, as you mentioned, and I write in our book Plague, when my grandfather died of lung cancer in the 70s, maybe 72 or 73, it was a closet disease. Nobody had anybody in the family with cancer. And so it was a hush-hush, let's not say anything. And, and then, the Cancer Institute that I worked at for more than 2 decades was started by then President Richard Nixon, when he declared a war on cancer and started right there at Fort Detrick in Frederick, Maryland. So, we've all seen the explosion, and there's something in our environment. And at the same time, we've seen the evolution of these viruses, these animal viruses, because they should stay in the animal, and yet we've been using them in our biological therapies and growing, entire fermenters like a craft brewing establishment, 200 liters or so of these animal cell lines. And we really didn't recognize, we appreciated for all these years the potential for this type of thing that we discovered in 2011 to happen. We thought it could happen, and in 2011, the big oh my God was, how many new viruses have we created, and how many new diseases? That was the big deal. That was it had to be shut up in hush hush because essentially everybody's at risk.
Yeah, I assume that this information to not have a widespread panic needed to be contained as quickly as possible. We'll be right back in just a little bit. You're listening to Health Made Radio. This is Dr. Michael Karlfeldt. Today's guest, I have Dr. Judy Mikovits. Welcome back to Health Made Radio. This is Dr. Michael Karlfeldt. I'm your host, and today I have Dr. Judy Mikovits. We've been talking about retroviruses, how we are exposed to them, what kind of impact they have on us, and literally I don't think we've seen the end of what they can do to us. And with this explosion of cancer, it actually, you can see these retroviruses are directly linked to the development of these cancer. And then also we have an explosion in things like autism, neurological diseases like MS, ALS, Parkinson's, Alzheimer's, dementia. All of these things are just exploding. And you think that they can be tied to these retroviruses as well?
Yes, essentially all of the diseases you just mentioned, autoimmune diseases, connective tissue disorders, lupus, Crohn's, Hashimoto's thyroiditis, Sjogren's syndrome, Behçet's disease, primary biliary cirrhosis, Ehlers-Danlos systems, all have been associated with retroviruses in one way or another over the past 3 decades.
So we pretty much unleashed all these modern-day diseases while we under the guise of protecting us from diseases like measles, right? Rubella.
Right.
And so we picked, instead of having measles, which when it has an outbreak, then maybe 6 or 7 or up to 10 people get it, and they consider that an outbreak. But here we were trading that for cancer, Parkinson's, ALS, and so forth.
Correct. But the good news is, and in fact, what we didn't recognize, but we now are starting to appreciate, that having these childhood infections like measles can actually protect us from retroviral-associated cancers like breast cancer, ovarian cancer, prostate cancer. So when you stimulate the immune system as a child against another family of RNA viruses, not retroviruses, you're actually developing a protective and a very real immunity that lasts a lifetime.
So it's almost like the childhood diseases existed just to kind of give your immune system a little bit of strength training to get a day at the gym to develop resistance against more serious conditions like cancer and autoimmune.
Correct.
That's amazing. And So you're talking about then, since these diseases are then linked to these retroviruses, to address these diseases and to shift them or to attack them, so to say, to use and the medication that was used for retroviruses would be the one of the better strategies, like using retrovirus medication or antiviral for cancer. That would be a feasible solution, correct?
Correct, and then those things are being done in multiple sclerosis, in Lou Gehrig's disease, ALS, in several others. Now we're using antiretroviral therapy as effective therapies. So when we address the retroviruses early in the treatment, remember the key with Magic Johnson is before the entire immune system is crippled, recognize the retroviral role, and you don't even need to test for it or just know the exposures or potential exposures. So early in the treatment of chronic illness, of other bacterial infections, remember I direct your audience to the movie or the book The Band Played On, because this was how, in the early days of HIV, we called it AIDS-related complex, we called it gay-related immune disease, we called it all kinds of things, but what it was these acquired immune deficiencies with Borrelia, with Mycoplasma, with Bartonella, with EBV, CMV, HHV6, parasites, heavy metal toxicities. All of these things go undetectable. And if you don't consider the retrovirus dysregulation of the immune system a component of it, then you won't be able to have your patients respond to treatment. But if you're always thinking, and your clinicians always think of a natural or plant-based therapy and consider the retroviral component, we can quickly revert these diseases into manageable things that we live with, just as we did with HIV AIDS.
And is there a test that people can take in order to be able to see whether they are impacted by these retroviruses?
I don't favor testing. I favor looking at the immune system, so I don't test for any given retrovirus. What I do is look for the immune markers, look for that dysregulated cytokine level, look for the chemokines, look for the inability of the natural killer cells to function, say CD56 or CD57. So yes, there are lots of immune response and immune mechanisms that are tested for every day in the cancer world. And so we can use these indirect measures of immune system, even a simple blood test. If you look at a simple blood test and look at some liver enzymes and look at the ability of the red blood cells, the sizes, the MCV, the MCHC. So we can look at oxygen carrying capacity and get indications of a retroviral involvement. So the testing that we do now, we can look at your white blood cells, we can look at your natural killer cells, we can look at your macrophages and your B cells, and see how they're functioning. And in many of these cases, since we've done this in cancer for 30 years of cancer biotherapy and immune therapy, there are treatments that exist right now to reestablish the homeostasis and balance that is dysregulated by the retroviruses.
Well, Dr. Mikovits, I truly appreciate the information that you've given, because I would consider this as fully life-saving, what you've just shared today, and I hope that all the listeners go and get her book called Plague by Dr. Judy Mikovits. It's an amazing book describing what we're exposed to. Thank you all for listening. It's all for today. I'm Dr. Michael Karlfeldt with HealthMade Radio. Remember to connect with us at healthmade.co. Health is what you make it.